Let's say you want to make a chemical in E. coli or yeast. The first question to ask is whether you know of any enzymatic pathway from a native metabolite to the product of interest. This is the pathway identification problem. The design synthesis problem in synthetic biology is akin to retrosynthetic analysis in chemistry or design synthesis in the area of electronic engineering, and looks a lot like program synthesis in computer science. For a chemical or activity that is already existent in nature, this is fairly straightforward. You just look up the paper that describes the pathway and build that. Suppose I have a large set of reactions that I know will spontaneously occur with the right enzyme and the right cofactors. How do I determine whether a chosen set of these enzymes will produce the desired product in a given cell? First, let's abstract these chemicals to A, B, C, and D to simplify this to just the logic without reference to structure. Provided any specific chemical, how do I know whether it can be reached by biosynthesis? If the chemical isn't present in any of the reactions, I have no basis for thinking it can be reached, so I should discard it. Our bag of reactions here only contains A, B, C, and D, so we can only consider those. Suppose we choose D. We can see that C can be converted to D, so if C is reachable, we can get to D. We also see that C is reachable if we can get to B, and B is reachable if we can get to A. We can't trace back from A any further through this data, but if A is already in the cell, then we should be able to make B, C, and or D by adding the appropriate enzymes. In short, we can define the reachability of a chemical by moving backwards through a bag of biochemical data and determining whether the reactants for each reaction are also reachable. If all substrates and cofactors for a reaction are reachable, then the products of the reaction are also reachable. This simple logic takes you pretty far in biosynthesis, but it's not quite perfect. First, the enzymes not only have to do the reaction, but they have to express and fold properly within your cell. Also, if the cell dies because there is a toxic intermediate in your pathway, then the product isn't going to be produced. We also haven't considered the effects of the other enzymes in the cell on these intermediates, the potential for our added enzymes to be post-translationally affected by other metabolites, and so forth. Finally, this all makes a really bad simplifying assumption about enzymes, that they only have one substrate. Just because an enzyme has never been observed to do a particular reaction, that does not mean it is unable to perform it. Because of this enumeration strategy will necessarily underpredict the actual list of reachable chemicals. Additionally, it will underpredict the unanticipated side reactions that will occur. With all these caveats stated, this basic logic is the good first step that gives rise to experimentally verifiable predictions. Let's look at a specific example so that you can see some of the complexity that you will run into in doing pathway identification. Shown here is the biosynthetic pathway for chloramphenicol and Streptomyces venezuelae. Within that first reaction sequence is the intermediate P-Amino phenylalanine. Suppose we wish to make that substance in E. coli. You'll notice that the route from shikimate, a molecule in all cells, to 4-amino phenyl acid is all worked out in this system. The last step, in which the alpha-keto acid is transaminated to the amino acid, is not explicitly stated. It's not clear what that enzyme is from the native host. Suppose we have the CMLB, CMLD, and CMLC reactions enumerated in our bag of data. We know that E. coli does indeed make charismatic acid and all the cofactors required for this sequence of reactions. Is P-Amino phenylalanine reachable? Logic would say no, but experience says yes. If we could just do the last step, yes, it would be reachable, but that reaction isn't observed to occur anywhere in E. coli because P-Amino phenyl acid intermediate is not spontaneously made in E. coli. E. coli does, however, make 20 proteinogenic amino acids. It does a very similar last step, transamination, for all these amino acids, and does so with only 3 enzymes, ASP-C, ILV-E, and TyrB. In fact, our substrate looks a lot like the precursor to tyrosine. They differ only by replacing the para-hydroxyl group with an amino group. Perhaps the enzyme that transaminates tyrosine, which is TyrB, will also accept our para-amino variant. If we go reading about TyrB, we'll find all sorts of database entries describing that TyrB likes the tyrosine intermediate. Here is its PubMed entry. We can get sequence info for it, and we can get homologs of it for a great many organisms. We can interpret the information that it likes the tyrosine precursor and that it likes a PLP cofactor. We can find ontologies that also describe this behavior, but nothing specific about P-Amino Phe. KEGG similarly doesn't get past describing the natural role of the enzyme. Brenda comes close. This database enumerates everything that a group of scientists could find in the literature and put into a database. They see from the literature that it likes tyrosine, tryptophan, 3-iodotyrosine, leucine, methionine, phenylalanine. It also has kinetic data for some amino acids, but nowhere in here is anything about para-aminophenylalanine. If you start digging into papers about TyrB, you'll find some more anecdotal or non-extractable knowledge. For example, TyrB even likes isoleucine as a substrate. It takes pretty much any alpha-keto acid that is a little greasy. Its specificity is very broad. These specificities manifest themselves as different kcat or over km values, primarily in the form of km. It's not a Boolean thing, though most other reactions will be undetectable. Databases don't currently capture this information. It is, in my view, the primary error in our modeling of biochemical systems that prevents us from making fine-grained predictions about what will happen when we add genes to cells. Fortunately, tools and technologies for addressing this problem are under active development.